Good day, everyone. Today, I will be presenting the major strategies used in gene therapy which aim to treat diseases caused by gene mutations. So, I will explain each strategy, how it works, and give examples so we can better understand the concepts. But first, let us answer why gene therapy is needed. Gene therapy treats diseases caused by defective or missing genes. It targets the root cause, and it can also provide long-term or permanent cure. So gene therapy focuses on the actual cause of genetic disorder or the faulty gene. Unlike normal treatments can only manage symptoms, gene therapy tries to correct the underlying problem. So that is how gene therapy works. So in gene therapy, there are four strategies. First is the gene augmentation therapy. Second is the targeted killing of cells. Third, the targeted inhibition of gene expression. And the last one is the targeted gene mutation correction. So these are four strategies fix genetic problems in different ways. So some add new genes, some silence harmful genes, and some kill dangerous cells, and some also repair the mutation directly. So let us tackle the first one, the gene augmentation therapy. So gene augmentation therapy or GAT, it introduces a healthy copy of a gene. So in gene augmentation therapy, we add a healthy version of a gene to replace one that is missing or not working. For example, this strategy is also used in genetic disorders like cystic fibrosis, where a faulty gene causes breathing problems. So we add a healthy gene that can improve lung function. And that is how gene augmentation works. And the process is you are just going to add a healthy gene through a package inside a harmless virus, and it will be delivered to the patient's cells. Once inside, the new gene produces a needed protein, allowing the cell to function normally again. The second one is the targeted killing of cells. So the targeted killing, you kill the diseased cells using toxic compounds or prodrugs. So this strategy destroys harmful cells, especially cancer cells, while leaving normal cells unharmed. So the process of targeted killing is to do this, a gene is delivered into the cancer cell. So when the patient takes a certain drug, that drug becomes toxic, but only inside the cancer cells, leading to their death, while the functioning cells is still unharmed. Targeted killing has two methods. First is the direct killing. So the direct killing, you produce toxic compounds. So the therapy makes the harmful cell produce a toxin that kills it. Just like, for example, in some experimental brain cancer treatments, a gene is added to cancer cells that makes them produce a poisonous enzyme. Once that enzyme is active, the cancer cells die. Next method is the assistive killing. In this method, a faulty gene will convert harmless drug into toxic ones. So in assistive killing, a gene gives harmful cells the ability to convert a harmless drug into a toxic form. One example is the gene used in certain cancer trials. Patients take a harmless drug called blasingclovir, and the cancer cells with the inserted gene will turn it into a toxic one, and the cancer cells will die. The third strategy is the targeted inhibition of gene expression. So it blocks expression of harmful genes. So this strategy stops a harmful gene from making a harmful protein. An example is Huntington's disease, where a mutant gene makes a toxic protein. Gene inhibition techniques can reduce the production of that toxic protein. There are also methods in targeted inhibition. First is the ASOs. So ASOs bind to mRNA and prevent translation. ASOs attach to the mRNA and block it from the cells. So, a real treatment that uses ASOS is Spinraza, a drug for spinal muscular atrophy. So, it helps correct the SMN protein levels by blocking faulty mRNA. The next method in targeted inhibition is the TFOs. So TFOs bind directly to DNA. So TFOs stop transcription by attaching to DNA before mRNA is even made. So this method is being explored for diseases like beta thalassemia, where the goal is to reduce harmful globin gene expression. Next is the RNA interference. RNA uses cRNAs to degrade mRNA. So, in RNA interference, small interfering RNAs destroy harmful mRNA. So, a real-life use of RNA is in treating hereditary amyloidosis. So, it is a liver disease caused by abnormal protein build-up. So, RNA reduces that protein production. The last strategy is the targeted gene mutation correction. So, gene mutation correction corrects defective genes. So, this strategy directly repairs the mutation in the gene. So, CRISPR gene editing is a good example. So, scientists uses CRISPR to correct mutations in diseases like sickle anemia, where a single DNA letter is wrong. So for the process is the defective gene is located, cut, and repaired. So the cell then uses the corrected DNA to make normal proteins again. So, a gene therapy provides a new solution to genetic disorders. So, in summary, gene therapy offers a powerful ways to treat genetic disorders by adding genes, killing harmful cells, blocking toxic proteins, or repairing mutations. These strategies gives us hope for treating diseases that once seemed incurable. So, thank you for listening.